We spend a lot of time talking about how proteins get made by ribosomes, but what if I told you that's just the very first chapter of the story? It turns out some of the most critical, most dramatic parts of a protein's life happen after it's been built. Oh, not by a long shot. That initial chain of amino acids that comes peeling off the ribosome, a lot of the time it's completely useless. Think of it like a rough draft, just a pile of raw materials that needs a ton of editing, tailoring, and finishing before it can actually do its job in the cell. So, what do we call all this work? Well, the official name is post-translational modification, or PTM for short. It's basically any change the cell makes to a protein after it's been translated from RNA. And that analogy of a finishing school, it's perfect. This is where a raw, clumsy polypeptide goes to become a sophisticated, fully functional protein. Right. It's exactly like getting one of those flat pack boxes of furniture. You've got all the wooden panels and a bag of screws, but you can't actually sit on it. Not yet. You have to fold it just right, cut off the extra bits, maybe add some finishing touches. A brand new protein is the exact same way. It needs to be folded, trimmed, and chemically modified to become the final working product. Okay, to really see this in action, let's follow the journey of a protein that's practically a celebrity. Insulin. We're going to trace its path from a simple, inactive chain into the powerful hormone that keeps our blood sugar in check. So take a look at this. This is the entire assembly line for insulin, all laid out. You can see it being built, then processed, and finally packaged up. It's a fantastic roadmap, and we're going to break this down step by step to see just how many modifications it really takes. It all kicks off right here, at the ribosome. The ribosome is chugging along, translating the genetic code into a long chain of amino acids. But here's the cool part. As it's being built, it's immediately threaded into this maze-like organelle called the endoplasmic reticulum, or the ER. This is where the real transformation begins. No. Once it's inside the ER, a couple of really crucial things happen. First, the chain starts folding up into its proper 3D shape. It doesn't just do this on its own. Other proteins help guide it. And second, an enzyme zips in and snips off a little piece at the very front called the signal peptide. You can think of this like tearing off the shipping label from a package. Its only job was to get the protein to the right address, the ER, and now it's not needed anymore. But we're still not done. After a trip through another organelle, the Golgi, the almost ready insulin gets packed into a little bubble called a vesicle. And it's only here, in the final moments, that a different set of enzymes, proteases, make these incredibly precise final cuts, removing a whole middle section. Only after these last snips is the mature, fully active insulin molecule finally ready for action. So we've seen how cutting parts away is a huge deal, but what about adding things on? Well, one of the most common and important modifications is basically adding decorations to a protein in the form of complex sugar molecules. This process is called glycosylation. Glycosylation is the cell's way of dressing up its proteins with these intricate sugar chains, which are called glycans. And let me tell you, these are not just for looks. These sugar additions are absolutely essential. They help the protein fold the right way, they make it more stable, and they often act as little flags or ID tags that other cells can recognize. And the cell has two main ways of doing this. It really just comes down to where on the protein it sticks the sugar. In what's called N-linked glycosylation, the sugar chain gets attached to a nitrogen atom on an asparagine. For O-linked glycosylation, it's attached to an oxygen atom on either a serine or a threonine, a different anchor point for a different type of decoration. And this image, wow, it shows you the chemical difference up close. Over on the left, you can see that N-linked sugar bonding with the asparagine. Then on the right, you see the O-linked sugar attached to serine. It's a tiny atomic level difference, but it can have huge consequences for what that protein does. And if you're thinking, okay, but do these tiny sugars really make a difference? Well, take a look at this. This is a lab technique where proteins are separated by size. On the left, that big, thick smear is a protein that's been heavily glycosylated. It's heavier because of all those sugars, so it moves slowly. On the right is the exact same protein, but stripped of its sugars. It's lighter, so it zips right down the gel. You can literally see the weight of these modifications. All right, so far we've talked about building proteins up and decorating them. But what happens when a protein is damaged, or maybe its job is just finished? The cell needs a way to take out the trash, right? And that brings us to an incredible system called ubiquitation. Ubiquitination is the cell's go-to tagging system. It works by attaching another, much smaller protein, called ubiquitin, onto a target protein. Now, this tag can mean a few different things, but its most famous job, well, it's basically a molecular death sentence. It marks the protein for complete destruction. Now, this tagging isn't random at all. It's a highly controlled, super efficient three-step cascade. First, an enzyme called E1 uses a burst of energy to activate a ubiquitin molecule. Then, that ubiquitin gets handed off to an E2 enzyme. And finally, a third enzyme, E3, acts like a scout. It finds the specific protein that needs to be tagged and attaches the ubiquitin to it. It's a beautiful, precise system. So, the cell has now slapped a Destroy Me sticker on a protein, but where does it actually go? Is there a cellular recycling plant that handles this kind of work? You bet there is. Meet the proteasome. This amazing molecular machine is the cell's protein shredder and recycling center all in one. It's this hollow, barrel-shaped structure that is specifically designed to recognize those ubiquitin tags, grab the doomed protein, pull it inside, and just chop it to pieces. Okay, let's walk through this because it's incredible. First, the chain of ubiquitin tags on the protein gets recognized by the cap of the proteasome. This cap then snips off the ubiquitin tags, they can be used again, and it unfolds the protein, feeding the straight chain into the central barrel. Inside this core, it's like a blender. A set of active enzymes just chops the protein into tiny little peptides, which are then spit out the other end, ready to be recycled by the cell into new proteins. So we've looked at cutting proteins, adding things to them, and tagging them for destruction. Let's just zoom out for a second and ask the big question. Why does all of this constant tweaking and modifying matter so much? Well, the short answer is that pretty much everything inside a cell depends on it. Without these finishing touches, hormones like insulin would never work, cells wouldn't be able to talk to each other, and worse, damaged and toxic proteins would just pile up until they killed the cell. It's absolutely essential. When you put it all together, the functions of PTMs are just staggering. They make sure proteins fold correctly. They let cells communicate. They switch on inactive proteins to create hormones. They are the cell's cleanup crew, and they basically create a level of molecular diversity that our genes alone could never achieve. And the level of control here is just breathtaking. It's not just that a protein gets tagged. As this quote points out, the way it's tagged, the type of chain, the pattern of attachment, is like a complex code. It's a sophisticated language that tells the cell exactly what to do with that protein and what its fate will be. So from the tiniest sugar molecule to a massive protein shredder, these tiny changes that happen after a protein is made are the true master regulators of the cell. Which leaves us with one final and really important thought. If this incredibly intricate system is the very foundation of a healthy cell, what happens when it breaks?